Davao River plays a very important role to the people of Davao City, considering the various services it provides, like in the sector of agriculture, tourism, and in households, to name a few. But unfortunately, the river's water quality showed constant deterioration as indicated on the 5-year water quality assessment made by the Environmental Management Bureau of Region 11. Based on the said assessment, the water contamination, particularly in the highly inhabited areas, is way beyond the acceptable level and is mostly obtained from the untreated domestic waste directly discharged to the river. A pleasant day to everyone. I am researcher Jana Michelle Aguilan, along with researchers Cyril Andre Javier and Pauline Mabitag of Davao City National High School, presenting to you the study entitled A Comparative Study of the Antibacterial Property and Flocculation Activity of Kangkong and Saluyot Leaves for Water Turbidity Removal. In the Philippines, about 5 million people rely on unsafe and unsustainable water sources, and 9 million do not have access to proper sanitation. The country is quickly urbanizing, and its expanding cities are struggling to provide appropriate water and sanitation facilities to new residents. The Davao River Basin is one of those that Davao City residents protect and value. Unfortunately, Davao River has been identified as one of the country's most contaminated waterways. Turbidity is one of the most common issues encountered during the water treatment process. Chemical-based coagulants, such as aluminum sulfate, are commonly employed in conventional water treatment processes to remove turbidity. However, despite their efficiency and cost-effectiveness, these chemical-based coagulants require pH and alkalinity changes in the majority of situations to adequately treat the water. In this study, we will be using plant-derived flocculants, kangkong and saluyot, which are polysaccharides in their natural state with a mucilaginous texture and a neutral pH, which are the main components needed in the flocculation process. The main goal of this proposed study is to gain knowledge about the antibacterial property and flocculating activity of kangkong and saluyot leaves extract in reducing turbidity count. In this, people can rely on non-toxic, cost-effective, and biodegradable flocculants in treating turbid water.
Here is a figure of the conceptual framework of the study. The independent variable is composed of two treatments. Treatment 1, 10 ml of kangkong leaves extract, and treatment 2, 10 ml of saluyot leaves extract. Both consist of 3 trials each. For the dependent variable, these are the parameters to be tested: the pH level, the nitrates and phosphate content in milligrams per liter, the total solid suspension in milligrams per liter as well, and the turbidity in nephelometric turbidity units or TNU. Now, let's discuss the methodology of the study. First, the preparation of the materials. Both kangkong and saluyot leaves will be collected from a Bini public market. Next, the leaves will be thoroughly washed with clean water to remove impurities. After air drying for 30 minutes using a mortar and pestle, 250 grams of kangkong leaves and 250 grams of saluyot leaves will be pounded separately. Lastly, 6 liters of untreated water sample will be collected from Davao River. For the preparation of the bioflocculant, the researchers will perform water extraction method to obtain the mucilage, which is the natural bioflocculant of the leaves. 250 grams of the pounded kangkong and saluyot leaves will be placed in two 1-liter beakers poured with 250 ml of boiled distilled water each after. It will be stirred for about 2 to 3 minutes to allow both kangkong and saluyot to react with the temperature of the water, ensuring their colloidal activation. Using a filtering media, both mixtures will be filtered and squeezed out by hand to obtain the extract.
Now let's take a look at the next step, the experimentation setup. First is to destabilize the colloids present in the water with the use of a coagulant. 1 liter of the untreated water from Davao River is going to be prepared and premixed with the coagulant, which is the potash alum, or commonly known as the commercially available tawas. As described in the study of Gomez et al. In 2017, an optimum dosage of 0.20 mg per liter of alum powder will be added to the water sample and will be aided after with the kangkong and saluyot leaves extract at a dosage of 10 ml. This will be done in triplicates for each treatment. The samples will then be set aside for about 30 minutes. The next step would be the parameter testing, where characterization of the supernatants, namely turbidity, pH level, nitrates and phosphates content, and total solid suspension, will be analyzed in Davao Analytical Lab. For the analysis of the antibacterial property, the samples will be brought to the Department of Science and Technology. For the data analysis, The statistical tool that will be used is the Mann-Whitney U-test since there are less than 7 trials in each treatment. The turbidity will be expressed in ephelometric turbidity units, while the nitrates and phosphates contents in the total solid suspension will all be expressed in milligram per liter. Percentage removal will be used in determining the parameters before and after the experimentation. Next is the risk and safety. Safety measures when conducting the experiments will be prioritized, such as the use of laboratory gowns and gloves. Lastly, the proper waste disposal, where the flocculated precipitate will be sun-dried and will be used as an organic fertilizer in one of the proponents' gardens. That would be all. Thank you. We are now open for questions and
I
recommendations.
will ask first the question to Edulan, our first presenter, or any of the team members may answer the question. Why do you think that, or why you considered these kangkong and these saluyot as the material that you are going to use, considering that these plants are edible and people are consuming these plants?
We have decided to use kangkong and saluyot, sir, because kangkong and saluyot has mucilage and cellulose, and these two are polysaccharides, which are also natural polymers that are used in flocculation. Kangkong contains 42.8% of cellulose, and saluyot has 64.4% of cellulose. And these natural polymers are also biodegradable and easy available from reproducible farms or forestry sources. That's why we have decided to use these two as our flocculants po.
Okay, so thank you so much. Anyone may answer the question. Why you decided to only measure 10 ml or to only test 10 ml of the property or of the material? Why not consider a higher concentration to test on its effectiveness in increasing turbidity and in decreasing the bacterial count? So why only 10 ml? Why not test the other concentration?
So based sa mga studies na naamo ang gibasean, sir, most of them only use 10 ml per 250 grams of water. So we decided to follow those, sir.
Thank you so much. What about in the number of replicates? How many replicates per treatment will you use? For example, for the test of pH for kangkong and for the test of pH for the saluyot, how many replicates will you accomplish?
Sa each parameter na gamiton namo is naami triplicates. So, na per treatment, naami tulo ka trials po, sir. So, for
Okay.
saluyot and kangkong, isa-isa sila na ay tulo ka treatment.
Okay, so what about, how will you know? How will you know or how will you confirm that the bacterial count or the pH level of the water decreased or increased in the conduct of the research study? Go ahead.
So we will test the parameters mentioned, sir, before and after the experimentation po.
Okay, very good. So you will have a testing of the parameters, your dependent variables before and after you apply or you treat your water sample. Where will you get your water sample and how many liters will be used for testing the effectiveness of the plant material?
In Davao River, sir.
Why you chose Davao River? Why not the other water sources?
Kasi po, sir, we have decided na sa Davao River may magkuha kay based sa water quality assessment na gibuhat sa EMB, ang water quality sa Davao River is gradually deteriorating po.
Okay, very good answer. I would like to, anyone can answer again the question, what about the disposal of the materials? How will you ensure that you have followed the strict protocols in the conduct of the disposal of these materials after you have implemented or tested the material to your water sample?
So, after po ng experimentation po namin, sir, yung precipitate is isan-dry po siya. Ang precipitate is isan-dry siya, and after that, it can be used as an organic fertilizer po, sir.
Sure na ba kayo na ang na makuha na material are really the material na gusto ninyong i-test? Baka these are not the materials na dapat na makuha from the plant sample. Meron ba kayong mga basis na after you have extracted it or get the property, sure na kayo na ito na yung gagamitin ninyo for the series of tests? Are any other confirmatory analysis na pwedeng gagawin?
We have not included po a confirmatory analysis in our paper po, sir. We are yet to find a source for that.
Okay, so you need to consider that one kasi during the conduct of the research, you are looking for the mucilaginous material. Am I right, Edulan?
Yes po, sir.
What's the property of a mucilage? How will you know that it's already the mucilaginous material? How sure are you that you have correctly extracted it?
Based sa amo na researcher, ang mucilage is mao na siya ang murag slimy material sa kangkong and saluyot.
Okay. What device will you use, Edulan, in the test of pH?
Sir, all the parameters, sir, including the pH level, the turbidity, the nutrient content, sir, will all be analyzed in Davao Analytical Lab po.
Okay, sige. Okay, so thank you so much and congratulations for the presentation. This is a promising paper. This is a very promising paper and I hope that we can accomplish